In this video, we'll take a look at the various means of physical control over microbial growth. I'm starting this presentation on the actual tables that try to detail for you the overall characteristics of that microbial growth, and we'll flip kind of back and forth between a few slides here that actually talk in greater detail about each one. The first category of physical control is going to be heat. In general, heat denatures proteins, and via protein denaturation, you can ultimately eliminate the ability of an organism to grow. If you think about proteins, in many cases they will also be enzymes, and by denaturing those enzymes, the metabolic processes that particular organism may be able to carry out will no longer occur. And without those particular metabolic processes, then the organism should not be able to grow. So, let's go back and kind of take a look here and talk a little bit more about the manner in which heat is used. Go back a little bit here. And like I've told you before, heat is a really useful means of microbial control. Typically, it works by denaturing proteins. We've already mentioned that. But there are different forms of heat that are utilized in order to try and maintain control. The first we'll mention is boiling. Boiling is effective, but it does have a drawback. The major drawback to boiling is that it does not destroy endospores. So boiling would not be a useful means of sterilization due to the fact that endospores could survive. Remember, in a previous lecture, we've talked about sterilization, and we mentioned that means the removal of all vegetative organisms, viruses, and endospores. Commercially, quite often we see pasteurization. This is a means of heat used to control microbial growth, and this usually involves using temperatures that are slightly higher than normal to try to decrease the overall number of pathogens. From a commercial standpoint, this means that, well, food and beverages would spoil at a slower rate. You can see you have high temperature, short time method, also known as HTST. This would require the heating of that particular beverage or product for a brief period of time at a high temperature in order to decrease that pathogen load. You can see milk usually is heated to 72 degrees Celsius for a very brief 15 seconds, and ice cream to 82 degrees for roughly 20 seconds. Now, it's not essential that you remember the timing for either milk or ice cream, but I do want you to keep in mind that this high temperature short time method, once again the HTST method, is a form of heat used to control growth of pathogens, and then keep in mind that it really falls up underneath that category of pasteurization. The ultra high temperature method, the UHT, is usually used for items that really don't require any sort of significant refrigeration, such as boxed juices and milk. It's also known as ultra pasteurization. This is where you use a very high temp for a much shorter time period, honestly, and then you cool it very rapidly. So you can see that for milk, ultra pasteurization, it requires 140 degrees, significantly higher than the 72, obviously, for only a few seconds versus 15 seconds. And then it's going to be rapidly cooled to try to prevent the loss of any flavor. When we work in the labs and in various medical facilities, you'll see that the preferred method of heat used to accomplish ultimately sterilization is going to be the autoclave. The autoclave is unique. This isn't just regular heat, it's heat combined with steam and pressure. So it's the pressurized steam heat that's typically used. Why is this more effective? Well, the pressure ultimately drives and allows for the penetration of the heat into the endospores, resulting in the death of the endospores. And once again, remember, for sterilization, we have to destroy the endospores. So typically, when we look at using an autoclave, most of the time it runs at 121 degrees Celsius with a pressure of 15 pounds per square inch for about 15 minutes. This can be variable. For larger volumes, say of liquids or larger quantities of materials that you may be trying to sterilize, it could be a longer time required. You can speed up that sterilization effort if you have a higher temperature, but most of the time 121 degrees is sufficient. Prions are thought to be destroyed if you utilize the autoclave at 132 degrees Celsius for an hour. Recall once again from a previous lecture, He said that in many cases, even when we perform sterilization techniques, the prions are thought to still remain. It is possible to try to destroy the prions, but like I said, this is a thought, so it's not 100% effective. Once again, the autoclave does use pressurized steam. If it's going to be effective, it does need to be able to enter that particular, the steam that is, enter that particular item that's being sterilized. So ultimately, we don't want to close containers very tightly if they're being sterilized. A lot of times if we're sterilizing flasks and things of that nature, we cover them loosely with a piece of foil. If it's some sort of test tube that has an attachable top, we'll loosen up that top in order to try to achieve that sterilization as well. Liquids in many cases, once again, we cover those but very loosely. And a lot of times we use tape that will react with the pressurized steam, and as it turns color, that would let us know that we have confirmed that the heat was able to penetrate our object and ultimately achieve sterilization. Next up, commercial canning. Commercial canning also utilizes heat in order to try and achieve control over microbial growth. The actual autoclave is called a retort. Not a major point for you guys to remember, but when you look at autoclaving in the commercial canning industry, we're really trying to destroy endospores of an organism called Clostridium botulinum. Clostridium botulinum spores grow under anaerobic conditions, and that's a perfect environment when you look at a canned food because there really is no air within a can. Otherwise, whatever you were canning would ultimately spoil very quickly. So to prevent the spoilage, the cans are airtight. Well, due to the fact that the can presents an anaerobic environment, this would ultimately allow for the thriving of those Clostridium botulinum spores. As they grow, they would produce botulinum toxin, which can be deadly if consumed by certain individuals, young infants, for instance. Technically, we consider canned foods to be commercially sterile. Now, this doesn't mean that they removed all the spores, but they removed the endospores to the point where they should not be a problem. You can see I say they don't necessarily remove all because there are some thermophilic organisms, those that really love high temperatures, whose endospores may survive. But if you look at it, that should not cause a problem for the human body because the human body is not a high temperature environment. Dry heat is much less effective than moist heat. You see, dry heat ultimately will require higher temperatures. Higher temperatures require more energy, they're more dangerous to work with, things like that. Now, we do use dry heat for sterilization under certain circumstances. In the labs, oftentimes you'll see what are called micro-incinerators. Those micro-incinerators have a heating element that stays at a very high temp for a prolonged period of time. Commercially, you might see hot air ovens and also some other larger ovens that can keep those higher temperatures for longer periods of time, like I said. But they're usually a little bit less effective because they require more energy input in order to use. This dry heat is really effective for metals, glass, things that aren't really heat sensitive or won't break down in the presence of that heat. Ultimately, they really have to be dry materials because any water would ultimately cause damage to the material that is within. Next up, we mentioned filtration. I'm actually going to go back to the table that we have here because we're kind of working through that. So we kind of covered the heat aspects. You can see boiling, pasteurization, autoclaving, pressurized steam, which we mentioned, incineration, dry heat ovens. Regardless of what type of heat, I would always tell you to keep in mind that heat ultimately, part of that destructive mechanism is that it denatures proteins. You see that mentioned here with dry heat and also with the moist heat. With filtration, we're going to take advantage of the size of the organisms. Typically what we see here is that some sort of membrane filter is used to eliminate organisms as materials pass through it. It could be that that material is air, it could be that it's some sort of liquid as well, but the filter has small pores that will allow the material to move through while keeping certain bacterial pathogens from moving through. We see really two major types of filtration. You've got the filtration of fluids. Once again, as I mentioned to you, we use a filter, variable sizes, but 0.2 micrometers is used to remove the bacteria. You see this often with beer and wine, commercial products, and some heat-sensitive medications. You may have one of these in your home, but HEPA filters are used typically for the filtration of air. The diameter is a little bit bigger, 0.3 micrometers. That's so that the airflow is a little bit easier, but nonetheless, you will still ultimately end up with filtration of a majority of the pathogens. Yes, some bacteria may get through because the diameter of the openings is a little bit greater, but nonetheless, still effective. Irradiation. This requires the usage of, waves of energy in order to ultimately try to damage the DNA of that particular pathogen. I'll go back to the slide which talks or discusses about radiation because I want to show you something here. Radiation occurs in different ways. Radio waves, microwaves, UV light, X-rays, and gamma rays. Multitude of different waves. Well, at the end of the day, the energy traveling in these waves is inversely proportional to the wavelength. What that really means is that the longer the wavelength, in other words, the higher the wavelength, the lower the energy. So you see here radio waves have a really long wavelength, but as far as their energy, their ionizing radiation, which is proportional to their energy, is really low. As you move toward the opposite end, notice when we get to the UV waves, we've got a shorter wavelength, but notice we've increased the amount of energy. So the shorter wavelengths have higher amounts of energy. You can see UV, X-ray, and gamma rays at the top end of that spectrum. Notice also that radiation would be bactericidal. We talked about that. These bactericidal agents would slow down the growth of bacteria. So really, when we look at trying to control microbial growth, not all forms of radiation are effective. Infrared, microwaves, and radio waves, not as effective, to be honest. You really, until you get into the UV spectrum and higher, that's where your effective radiation techniques will come in. Talking a little bit more about that irradiation, the gamma rays and the X-rays are the highest energy waves. We just saw that a moment ago. They're going to work by destroying DNA. They can also react with oxygen in order to produce oxygen radicals. These oxygen-free radicals can be toxic to any anaerobic organisms that might not be able to process them. Not as damaging to aerobic organisms because aerobic organisms naturally have enzymes to break down oxygen-free radicals. You can use the gamma rays to sterilize heat-sensitive materials, and in fact, some food products are irradiated with gamma rays in order to decrease the pathogen amount within them. But that's kind of a debatable topic because at the end of the day, a lot of people don't want their food irradiated. UV radiation is commonly used in commercial kitchens. If you've ever been around a kitchen, you may have seen a UV light box. It's not a really great means of control, but nonetheless it helps because at the end of the day, the UV radiation can't penetrate surfaces very well, but it can be useful for organisms in the air and on top of those surfaces. So if we were trying to reduce or control pathogens within some material, UV radiation would not be effective at all. Microwaves, they're mentioned here because while they're not individually strong enough, if you go back to this image here, not very strong, but as they penetrate an object, they can generate heat. And the heat ultimately can be lethal. But you can see here, they don't heat food evenly, so it's possible for some organisms to survive. Finally, here we have high pressure processing. This requires the use of pressure in order to try and destroy microbes. You see this a lot commercially with foods especially because even though the food is placed under high pressure where the organisms can't survive, the food doesn't lose any of its natural taste or flavor, which is obviously something that the companies producing that particular food item would want. So this high pressure processing is useful in processing foods that would be available commercially packaged. For instance, like guacamole is what we usually refer to here, and juices as well. So that kind of concludes the discussion about the physical methods of control. What I would tell you all to definitely go back and take a look at are the various methods of physical control along with how they work. For example, irradiation being able to damage or destroy DNA. And then you definitely want to keep in mind some uses of those various methods of control. For instance, like with the high pressure, notice how we're using that in order to destroy or decrease the amount of pathogens within packaged foods. That's a little bit different than when you look at the heat, which is used in order to achieve control for the canning processes. So kind of keep that in mind. Different forms for different processes.